exactly these thread-like structures in the cell work? Understand. So, for example, we have this one thread, right? It looks like a single thread. It looks like a single thread. Yes or no? So for me, it is a like, or it is a like, one sec. It is like this cell. Yes or no? Yes or no?
Yeah.
But when you sort of elaborate it, for me, it looks like actually a strand. This double-sided strand. Are you understanding?
Yeah.
And you understand that every portion, so this is obviously the connecting point here. So there are two strands, you understand. One strand comes from mother and one strand comes from father. Are you understanding my point?
Yeah.
Huh? So now let's suppose there are certain characteristics. Let's suppose your father's hair are curly, your mother's hair are straight. So this portion of this entire thread will hold information regarding your hair shape. Okay? Let's suppose I say that, this another next portion, this stores information about your mother's eye color is, let's suppose, black, and your father's eye color is, let's suppose, brown. Then another portion will hold information, and in every portion 10 informations are stored. Okay, like that. 10 to 15, 20 informations are stored. So there could be another possibility that this portion, this much portion. So this strand holds information about the shape of your nose, shape of your father's nose, and this part holds information about the shape of the mother's nose. Are you understanding? Okay?
Yeah.
Are you understanding?
Yeah. Yeah. Got it.
अरे मुझे लगा मामा जी थे, मैंने इसलिए बंद कर दिया. मैं खोल रही हूँ, बंद है ऊपर से, रुको 1 मिनट. आ जाओ ना बाहर. अच्छा, सॉरी, 1 सेकेंड, 1 सेकेंड. All right. So now understand what exactly happens. So this strand basically is carrying information. Every strand, every part of the strand is carrying certain information. One from the father and one from the mother. Do you understand my point?
Yeah.
Right? This information, this part of the strand is called as the gene. Gene. So I hope you can understand that this might have lots and lots of genes over it. Yes or no?
Yeah.
These are nothing but lots and lots of genes right over it. Any doubts on this? So this is one
Yeah.
gene, this is another gene, this is another gene, this is another gene, another gene, another gene, another gene. So what do we say? Genes are the structural and functional unit of the nucleus, and it carries all the hereditary informations. I hope you're understanding.
Yeah.
Yes or no? It carries all the hereditary information. Understood
Yeah.
or not? Right? So
Got
it
it.
carries all the hereditary information. So there are lots and lots of genes on the particular chromosome or on the particular chromatin material, because there are 23 such threads. Yes or no?
Yeah.
Yes or no? If there are 23 such threads, what would that mean? Tell me. Tell me. So that is the concept. I hope you're understanding, right?
Yeah.
Any doubts in this?
No, none.
Okay. All right. So one strand, I hope you're understanding, coming from mother, one strand coming from father. They get entangled, and that's how it is present in us. So if you talk about your mother's cell, in your mother's body cells, all what would happen? They would also have two strands. So one strand would come from the nana, your nana, so as to understand, and nani. That means your mother's mother and your mother's father. Are you understanding?
Yeah.
So now when this kind of strand is actually dividing to form your strand, because you have gained the strands from your mother, but your mother's strand was actually one of the strands of your mother was actually coming from nana or nani. I hope you're understanding.
Yeah.
So there might be a possibility that you might inherit certain properties of your nana or nani.
Okay.
Yes
Yeah.
or no?
Yeah.
Yeah. So that's how it is. So basically, you get the properties of your nana, per nana, great grand nana, and so on. It's like you can have the possibility of, inheriting the characteristics of your ancestors. Lots and lots of ancestors, like, two people I can name them, great grand ancestors, and even beyond that. Because gradually and eventually, we are getting these strands from them only. I hope you're understanding.
Yeah.
Right? So this is the logic. Now, how are these strands basically joined together? That is the case. That is the important point to understand actually. So just understand what exactly it happens. One second. Yeah. So now how is it? Just have a look. So you understand. When we sort of draw about this particular strand, okay? So this strand is actually, made up of lots and lots of substances. So one is what? One is a sugar molecule. And there is this phosphate group, okay, right? And this group forms nucleoside. Then another sugar molecule and another phosphate group. So this group of sugar molecule and phosphate group is again called as another nucleoside. It's called as another nucleoside. And these nucleosides are joined together by a group which is called as nucleotide, is joined by proteins. So three things make up this one strand. So if I take out this one strand is actually made up of sugar molecule, is actually made up of phosphate group, and is actually made up of protein. Are you understanding?
Yeah.
All right. So these three things make up this one strand, right? So you're not supposed to get into a lot of detail as to what is the sugar molecule and stuff, but I'll tell you major portions of it, right? Because that is necessary to understand. If I don't teach you that, maybe your teachers teach you or don't teach you, but this is important to understand. So you understand that
Yeah.
this nucleoside, when two nucleosides are connected by protein, so this chain is formed. Okay. So there are different types of protein that you find here. You find, right? So we have purines. So proteins are of two types. So proteins are of two types: purines and pyrimidines. Purines and pyrimidines. Okay. Purines again are of two types. One is called uracil. Sorry, not uracil. Purines are of two types. One is guanine and adenine, right? And pyrimidines are of two types, right? That is thiamine, cytosine, and uracil. So these are the protein types, basically. All right. Are you understanding?
Yeah.
So what is the job of this protein? The job of this protein is to actually connect this nucleoside with another nucleoside. To connect one nucleoside with another nucleoside. All right.
Yeah. Got it.
Now understand. So these are the basic protein. Now, what is the basic difference between them? Purines are generally single. In the structural. That's a structural difference. So the structurally they have one ring, and pyrimidines would have thereby two rings. That's the difference. That's the only difference, right? So that's what you're supposed to remember. But in detail, if I'll go, then I can be able to do the structures also. But that is not of no use for you. You are in grade 9, right, Sonia?
Grade 10.
Grade 10. Yeah. So this is not of much use for you. For now, you just understand that this is the basic structure, right? Now, understand. Which sugar molecule do we have in here? We have deoxyribose sugar. Deoxyribose sugar. We have deoxyribose sugar. That's the name of the sugar molecule that we have. All right.
Yeah.
Now, just have a look here. Just have a look. Now, just see. So, when I'm looking at this particular chain, obviously, this one chain is independent chain. This blue chain is also an independent chain. So obviously something has to hold them together, isn't it? Are you understanding?
Yeah.
So what is exactly happening? Understand. So you understand there are lots and lots of bases here. A, T, C, G, G, C. And why are these bases coming out? Because these bases are sort of connecting the sugar molecule and the phosphate group. That is the nucleoside they're holding. Right? Then I'll say again A,
Yeah.
then T, then C, then G, then G, then C, then T, then T, then T, then A. So there is a certain pattern, and this pattern of protein group is different in everybody else. You understand? So I hope you are understanding that how many patterns this could make, because it is without repetition, it is repeating itself. It could be A, right? It could be T, it could be C, it could be G, it could be G, it could be C, it could be T. I hope you're understanding.
Yeah.
And here also, what is happening? It is going to be T, G. Now, understand what am I doing now? Now, there is a rule for this. Always you understand in front of A, if on one strand there is A, adenine, in the other strand just in front of adenine would be thiamine, AT. Because A and T can join with itself. And C can join with G. C is what? Cytosine. And G is what? Guanine. T is what? Thiamine. And A is what? Adenine. Are you understanding? So these are the nitrogenous
Yeah.
bases. So if on one particular strand adenine is present, just right in front of it we will have thiamine always. Always. Right in front of T we will always have adenine. So thereby they are able to join. And when they're able to join, I hope you are understanding the strands are able to hold together. You understand? The strands are able to hold together. Any doubts in this? Yes or no?
No. Got it. Yeah.
Right? So C and G, right in front of C would be G. Right in front of G would be C. Right in front of G would be C. Right in front of C would be G. Right in front of A would be T. A, C, G, G, C, C, G, A, 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 T, T, A, G, C, C, G, A. Are you understanding?
Yeah.
Any doubts in this? Just tell me.
No,
So,
none.
this is called complementary pairing. This is called complementary pairing. Any doubts in this? Yes or no?
None.
What do you mean by complementary pairing? Tell me that. That A shall always combine with thymine. Adenine shall always combine with thymine. Cytosine shall always combine with guanine. That's the complementary pairing, which holds the two strands together, thereby making this double-stranded chromosome. Double-stranded chromatid material. Yes or no?
Yeah.
Any doubts in this? Just tell me.
No, none.
All right. Now that is the concept,
Yeah.
right? Now, at the time of cell division, what exactly happens? Just understand, right? Now, what exactly happens? Just have a look. At the time of cell division, what exactly happens? This double strand, decides to open because of certain enzymes being released. So these double strands open up. So I hope you're understanding that there was A, T, G, C. Yes or no? So if I say the
Yeah.
entire if the entire DNA doesn't open up, at least a part of it decides to open up like this. And there might be certain strands here. A, T, C, G, 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 C, T, A, G, C, T. So what do we have in here? Right in front of A, it would be what? Right in front of A, it would have been
T.
T. Right in front of T, it would have
Yeah.
been A. In front of C, it would be G. In G, it
Yeah.
would be C. It would be C. It would be C. It would be G. In front of T, it would be A. In front of A, it would be T. In front of G, it would be C. In C, it would be G. Do you understand? In front of T, in front of C, it would be G. In T, in front of it would be A, isn't it?
Yeah.
Now, what would that mean? Just tell me. So it says at the time of cell division, definitely. At the time of cell division, definitely, this reopens. And now a new duplicate of it is formed. A new duplicate of it is formed. I hope you're understanding. So a new chain, because of all the enzymes being produced, because of all the enzymes being produced in the you call what do you call, all the enzymes being sort of produced in the nucleus. I hope you're understanding that this is going to be a new chain is going to be produced. This new chain starts to produce like this. And how are they held together? So new enzymes are definitely being produced in the nucleus itself. So right in front of A would be T, in front of T shall be A, G, C, C, G, C, C, G, A, T, C, G, A. Are you understanding my point? Now just have a look here. Don't you think that this particular chain would exactly be looking like this? Right in front of T would have been A here, isn't it? So do you agree that in both the chains AT is same, TATA is same,
Yeah.
CG is same? So this is called duplication. This is called replication. This is called replication. So what exactly happens? The chromosome braid basically opens up. The chromosome braid basically opens up. Yes or no? And when the chromosome braid actually opens up, what exactly means? Tell me. Tell me. It duplicates itself. It replicates itself. Are you understanding?
Yeah.
Just tell me. Right? And when it replicates itself, when it replicates itself, so two new strands are actually formed. So at the time of cell division, I hope you're understanding what is happening. That the nucleolus, basically, the cell basically was small. The cell grows up in size. The chromatin material, it becomes into the rod-like structures. And when the chromatin material acquires these rod-like structures, so it becomes a chromosome at the time of cell division. I hope you're understanding, right?
Yeah.
And we had this particular cytoplasm outside, and the cell membrane decides to, disappear. So there are these chromosomes. These are these chromosomes lying in the cytoplasm freely now, because the entire cytoplasm, entire nucleoplasm has been, the nuclear membrane has been dissolved, has disappeared. You understand? And we have this particular
Yeah.
nucleolus. We have this particular nucleolus that is also disappeared. That is also disappeared. Are you understanding my point?
Yeah.
Hmm? And we would also say one thing, that every cytoplasm has now duplicated itself. Every cytoplasm has now duplicated itself. So these are the chromosomes. So how many chromosomes do you think we had? 23. Every human has 23. So this blue part that I'm drawing. So if this was my original chromosome, which is double stranded, so now we have the duplicate copy of this as well. Do you understand? This would be
Yeah.
the duplicate copy of this as well. Any doubts in this?
No, none.
Any doubts in this? Tell me just that. All right.
Yeah.
And duplicating means, basically, if I look into the details, I hope you're understanding. There are two strands in the blue one. There are two strands in the blue one, and there are two strands in the red one. So effectively, in this part, there are four strands. Yes or no?
Yeah.
Hmm? So we'll do up till this part only. Tomorrow, if you want, we can connect. Tomorrow, can we connect?
I'll let you know.
Okay, sure. All right. Tomorrow is Sunday. Do we
Okay.
do we have a class on Sundays generally? No, we didn't decide for
Uh,
Sundays classes.
and we didn't decide.
Yeah, that's fine. No worries. So just let me know, right?
Yeah. Okay.
All right. Bye.
All right. Thank you.
Bye.
Yeah.
Bye.
Bye.